Hello, everyone, and welcome to today's webinar titled LDLR-Related Receptors are Evolutionary Conserved Cellular Receptors for Divergent Alphaviruses. This webinar is part of the Microbiology Virtual Week, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. Now, you can find that live chat located at the right of your screen. You can also participate by submitting as many questions as you want during this presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Now, if you have any trouble seeing or hearing this presentation, just click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located at the abstract window below the presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Jonathan Abraham, Assistant Professor of Microbiology at Harvard Medical School. For a complete biography of our speakers, please visit the Biography tab at the left of your screen. Dr. Abraham, welcome. You may now begin your presentation, sir.
Some of our labs work. Thank you for the opportunity to tell you about some of our labs work. So the title of my presentation is LDLR-Related Receptors or Evolutionary Conserved Cellular Receptors for Divergent Alphaviruses. And so we'll start off with some background on alphaviruses. These are arthropore-borne enveloped RNA viruses that fall into two main groups, Old World and New World. And this is based on their serology and geographic distribution. They tend to, when they infect humans, cause two types of illnesses, either fever, rash, and arthritis, or encephalitis, and by that I mean infection of the brain. And the case fatality rate is particularly high for the alphaviruses that cause encephalitis. And so to give you an example of one of these viruses that some of you might have heard of, more featured in the news back in 2019 is Eastern equine encephalitis virus. This virus caused outbreak in 2019 before the COVID-19 pandemic that affected about 34 people in 7 different states. So the case fatality rate for this infection is quite high. About 50% of the people who get infected can die from infection. And those that survive, unfortunately, end up, in many cases, with permanent neurological sequelae, which means that they require institutionalized care thereafter. So the burden of this illness on healthcare can be quite high, particularly should this virus cause a pandemic. And so Eastern equine encephalitis virus, here I'm showing you an example of how this virus is amplified and maintained in nature. The virus can be carried by mosquitoes, where it replicates and ultimately via blood meal can infect the bird, in this case serving as a reservoir. Birds can then infect additional mosquitoes, so the cycle can be effectively a reservoir in nature for the virus to be amplified. But then when a bridge vector here shown on the right part of the slide is able to get a blood meal from both a bird, but then a dead end host, including humans or horses, this is where you get highly lethal disease. And I should note that there are actually no vaccines or drugs against alphavirus infection. And so the structural protein is subdivided into 6 polypeptides as the protein matures. There's an N-terminal capsid, envelope glycoprotein fragments E3, E1, E2, and 6K. E1 and E2 are really the business end I'll be telling you about today during my presentation. E3 is a chaperone for E1 and E2, and E1 and E2 associate as dimers of trimers that form icosahedral shell on the surface of the virus, which is shown on the right part of the slide here. So you will see there that there's a outer layer in this case shown in blue and an inner layer shown in orange. So these are the trimers of the E2, E1 glycoproteins. So classically, it's been thought that E2 has most of the ability to bind cellular receptors, and E1 is what mediates fusion of the viral and host cell membranes after viral particles are internalized into acidified endosomes. Although some structural analysis recently suggests that both E1 and E2 actually are both participating in receptor binding. And so we started this project in our lab back in 2018. At the time, not much was known about cellular receptors that allowed alphaviruses to enter cells. That being said, the advent of CRISPR-Cas9 screens and the robust genetic approach this offered to find receptors for viruses meant that multiple labs had been working on identifying receptors for these viruses. And so in this case, you can see that for the virus the virus chikungunya virus, MXRA8 could serve as a cellular receptor. My apologies about that. So MXRA8 is a matrix remodeling associated protein that plays a poorly described role in the biology of joints. And so in this case, the lab of Mike Diamond with collaborators, they were able to find that MXRA8 associates with the viral spike protein by binding a cleft that is formed by E1 and E2. In this case, you can see that the E1 protein is shown in orange and the E2 glycoprotein is shown in blue. MXRA8 is the molecule on the right side of the slide that is shown in purple. And so we set out to use CRISPR-Cas9 screens to identify receptors for some of the encephalitic alphaviruses, because at the time, no cellular receptors were known for these viruses. So a lot of these agents are actually BSL-3 pathogens, and so in order to be able to study them safely, what we had to do is develop a system that allowed us to have effectively the ability to pseudotype the virus so that it enters cells, but could then not replicate to spread to infect other cells. And the way we did this is we took the genome for an alphavirus that's actually a BSL-2 pathogen, Ross River virus. In this case, you could see that the genome is subdivided into 4 non-structural proteins, NSP1, 2, 3, and 4, as well as the structural proteins, in this case, capsid E3, E2, 6K, TF, and E1. Hopefully, you can see then that as a strategy, what we're able to do is split this genome into 2 segments, one encoding a plasmid that has the genome, but whereby the glycoprotein has been deleted and replaced by a reporter that is downstream of a 2A peptide. So in this case, the reporter is GFP, if we want to do FACS to monitor viral entry, or the reporter could be CD20. As I'll tell you on the next slide, we could use that as a marker of a cell being infected, but being able to track cell surface expression allows us then to take infected cells and deplete them using beads against that marker. And so this plasmid 2 here, in this case, could be, instead of the Ross River envelope glycoprotein, could be the glycoprotein for Eastern equine encephalitis virus, or could be the glycoprotein for additional alphaviruses we were interested in studying. And so we use this system to design a CRISPR-Cas9 screen that's shown on this slide. In this case, we have guide RNAs that are transduced into cells. This is followed by pure myosin selection. Once pure myosin selection happens, all cells should have, the way we set up the experiment, a single guide targeting an individual protein. We take these cells, we challenge them with RVPs, or reporter virus particles, that express CD20, being again this cell surface marker. In this case, what you'll see is that I'm showing you as these little yellow marks on the cells that are infected with CD20 RVPs, effectively cell surface expression of this marker. And then we use magnetic beads to deplete the CD20 positive cells, so that only cells that are resistant, meaning cells that contain guides that knocked out potential receptor candidates, are carried through. And so to increase the signal to noise of the assay, we can do this multiple times. And at the end, we use next generation sequencing to quantify the both relative prevalence of guides, as well as the total amount of reads per guide using next-gen sequencing. So another trick we used here is that to only focus on proteins that would probably be cell surface receptors, we did not do a genome-wide screen, but instead designed our screen so that each guide would only target proteins that are suspected or known to be membrane-associated. So this meant that the total genes we were targeting were about only 4,400 genes in total. And because of the way the library could be designed at the time, it also meant that we could use more guides per gene. So almost genome-wide screens have 6 to 8 guides per gene. We had 10 guides per membrane-associated protein in this assay. And so we first validated the screen using viruses that had known cellular receptors, including Lassa virus, which is an arenavirus, and Lujo virus, which is another arenavirus. And so we were quite confident that this approach could yield very robust results. And so when we carried out the screen with Semliki Forest virus, which is an old world Alphavirus that, interestingly enough, has been reported to cause a case of encephalitis in humans once, but is known to cause encephalitis in animals, including neonatal mice, as well as horses. The top hit in the screen here is shown with an RRA score, which is based on the bioinformatic pipeline related to how often the guide is present in that final population I was telling you about on the last slide, from the next-gen sequencing results, as well as how many guides per gene are found. 10 out of 10 guides were found for VLDLR, and there were very high read counts for that guide. And VLDLR stands for the very low density lipoprotein related receptor. It is a type 1 transmembrane glycoprotein that has a central job of bringing into cells the very low density lipoprotein. It's also expressed in the central nervous system, where it is a receptor for relin, and this molecule, along with relin, ultimately helps orchestrate the laminated structures that occur in the developing brain of mammals. And so a second hit on the screen was HSP90B1. Interestingly enough, this protein is the chaperone for another protein called PCSK9, which is a master regulator of LDLR-related receptors. Then SCD3A was the only other statistically significant hit. This is a protein that is involved in the replication of other positive sense RNA viruses, flaviviruses, but had not been known to be implicated in the entry or replication of alphaviruses. And so everything downstream of this now, including the CHRM1, these were actually within the range that we knew based on our pilot studies would actually not be statistically significant or relevant. So we need to really focus in on VLDLR. I should add that VLDLR is also a receptor for other viruses, including minor group human rhinoviruses, as well as vesicular stomatitis virus. So the fact that it was also a receptor for other viruses made us even more confident that it would probably be playing a real role in the entry of this particular alphavirus. And so the usual experiment we do to validate any receptor is to take wild-type cells, in this case hector T cells, knock out the receptor using specific guides this time, targeting the receptor only, and then showing that rescuing expression of the receptor using cDNA encoding VLDLR could actually lead to robust entry in this case. So correcting for the defect that was seen by individual knockout of the target gene. And so an additional experiment we wanted to do, because Semliki Forest virus, interestingly enough, is known to infect multiple subtypes. This has actually made it a candidate for gene therapy as well as vaccines, because it's been known to affect multiple cell types. So we wanted to know whether or not this ability to use VLDLR was related to the tissue tropism of this virus. So if you see in this case, if we take cells treated with no antibody, an anti-VLDLR antibody, or an anti-HLA antibody being their negative control, you could see that we could block entry into 293 T cells. And then when we tried cells derived from lung, liver, lymphoid, and bone, we had similar results. Bone was interesting because for those cell types, we saw only about 40 to 50% reduction in entry, suggesting to us that perhaps there might be alternate receptors on this cell type or alternate pathways of entry. And then lastly, the most relevant cell type would be neurons. So these are two immortalized neuronal cell lines. We saw that an antibody against VLDLR again could robustly block the entry of Semliki Forest virus. And so the next set of experiments we wanted to do was to define exactly where the virus might be attaching onto the receptor. And so the receptor is organized, again, as a type 1 transmembrane protein. I'm not showing here the cytoplasmic tail, but it would be below the cell membrane here. The N-terminal portions of the receptor here, which are most membrane distal, have a series of cysteine-rich modules called LDLR class A repeats, or LA repeats, that are arranged in tandem here and vary slightly from each other in sequence, but all coordinate a calcium and ultimately are what comprise a ligand-binding domain that is what binds to VLDL and relin. And then in red and in green, I'm showing you the EGF-like module that is not involved in receptor in ligand binding, as well as an O-linked domain here that has O-linked sugars in yellow. And so to map where the virus might bind, we decided to generate two additional constructs where we either deleted the ligand binding domain or deleted the EGF-like domain and kept the ligand binding domain intact, and repeated these expression experiments in K562 cells, which are cells we found were highly resistant to infection, to see whether or not we'd be able to define exactly where the virus was binding. And so what you'll see here in this case is we have wild-type K562 cells. I should add that these are chronic myelogenous leukemia-derived cell line that's poorly differentiated. And whatever alphavirus we've thrown at them, usually there's very little to no background entry. So this is what you can see here with the wild-type K562 cells. There are no GFP positive cells when we challenge these cells with Semliki Forest virus, RVP. The VLDLR ortholog, human ortholog, when expressed in these cells, allowed for robust entry. Once we deleted the ligand binding domain, that's the next column over, we saw loss of entry. When we deleted the EGF-like domain, but not the ligand binding domain, we saw that entry was restored. And the NRP2 flag in this case is a negative control receptor. This is a receptor for Lujo virus, unrelated virus. And so this suggested to us that the virus was actually associating with the ligand binding domain that contains these cysteine-rich LDLR class A repeats. And so we sort of then started to think really hard about the implications of this, because this was the first time an LDLR related member was identified as a receptor for an alphavirus at the time we were doing these studies. And I think when one is looking at a family of viruses that are related to each other and perhaps cause similar pathophysiology, I didn't mention to you there's Venezuelan equine encephalitis virus that also causes encephalitis in humans and horses. I mentioned to you Eastern equine encephalitis. There's also Western equine encephalitis. So all these viruses that have related E1 and E2 glycoproteins. So one of the first questions we wanted to answer is whether or not VLDLR was a receptor only for Semliki Forest virus, or might be a receptor for some of these other alphaviruses, including Old World and New World alphaviruses. Another thing I should add is, at the functional level, VLDLR has a close homologue in the brain called ApoER2, which is also a receptor for relin. And so VLDLR and ApoER2 are very closely overlapping in their tissue distribution in the brain, and their function as relin receptors important in neural development. So the next set of experiments, we'll show you only the ones that worked. We tried to see if we took K552 cells, SFV or EEV or SINV, the related Old World alphaviruses, on K552 cells, here in this case expressing MXRA8, the chikungunya receptor. We saw no entry, but when we expressed VLDLR or two isoforms of ApoER2, all of a sudden we saw very robust entry. I'm not showing you this here, but VLDLR and ApoER2 are also related to LDLR, which is LDL receptor, effectively. That's perhaps the best studied member of the family. So LDLR did not work, but VLDLR and ApoER2 did function as receptors for these three alphaviruses. We tested additional alphaviruses and saw no effect on entry. And so I think a key important aspect of whenever a receptor is identified for a virus family is to demonstrate an interaction between a recombinant protein and the viral glycoprotein, or recombinant receptor fragment, I should say, and the viral protein. And the way we did these assays is we took Q93 T cells transfected with the E2, E1 envelope glycoproteins, and then stained these cells with Fc fusion proteins that either contain the VLDLR ligand-binding domain fused to an Fc segment, so that we can stain with anti-Fc antibodies to detect cell surface binding of these constructs, or MXRA8 in this case, which are a control protein, which is a receptor for chikungunya virus, as I mentioned, identified by Mike Diamond's group over at Washington University in St. Louis. And so this is a control here, cells transfected with chikungunya E2, E1 will actually bind the MXRA8 Fc fusion protein, but not the VLDLR-Fc fusion protein. But when we did a similar experiment, but instead with Q93 T cells overexpressing Semliki Forest, Eastern equine encephalitis, or SINV viruses, we saw the reverse effect whereby the VLDLR-Fc fusion protein was binding to cells, but not the MXRA8 Fc fusion protein. So this suggested to us that indeed our Fc fusion proteins containing the ligand binding domain for VLDLR was the molecular determinant that allowed direct binding to the envelope protein. But I think one aspect of LDLR receptors is that they, of course, bind lipoproteins. And so the experiments we had done up until this point did not confirm that the association between the envelope glycoprotein and the ligand binding domain was a direct one. You can imagine that even in these binding studies, if our LBD-Fc fusion protein is able to bind lipoproteins in serum, and that complex then associates with E2, E1, that we'd still see binding. I'll give you an example that VLDLR has been implicated in cellular entry of hepatitis C virus, but for that virus, for example, it's thought to be actually an indirect association with lipoproteins that are decorating the viral particles. So all that being said, we wanted to try to use as pure as possible preparations to demonstrate a direct interaction between the VLDLR-LBD and the E2, E1 proteins of alphaviruses. And so the way we did these assays is we first relied on a system that allowed us to make virus-like particles. So these are particles that, again, recapitulate the icosahedral symmetry of the actual virus and can simply be produced by transfecting in the structural proteins under a CMV promoter into cells. And so we were able to get these very nice virus-like particles. The scale bar here is about 100 nanometers. The virus is supposed to be 70 nanometers. And we examined these under the electron microscope. I did not see evidence of associated lipoproteins. We also took these particles that were purified and submitted them to mass spec, where we did not identify any evidence of associated lipoprotein peptides. So this secured to us that we had very pure preparations. And so for binding assays, we use a technique called biolayer interferometry. In this case, what we have is a sensor tip that is dipped into a solution that allows them to capture either VLDLR or ApoE2 Fc fusion proteins, or the control MXRA8 ectodomain. And so when these sensor tips bind to first this ligand, and then they're dipped into a separate solution that contains an analyte, in this case virus-like particles, you would see binding. But the way this assay was done, which is really a competition assay, we first pre-dip the tips into buffer and then VLPs. So with under the VLDLR graph, the left side of the graph, you'll see that when the VLDLR coated tips were dipped into buffer and then VLPs, we saw a response on the y-axis suggestive of binding. Receptor-associated protein is a ligand antagonist. So when we pre-dipped VLDLR into RAP-containing solution and then did the binding assay, we saw a decrease of binding to VLPs. When we pre-dipped the VLDLR-coated tips into solution containing transferrin, we did not see a decrease in signal. Transferrin here was our negative control. And you can see at the next set of bar columns here with buffer RAP and transferrin for ApoE2 ISO1, we also saw a decrease in binding with RAP. So this allowed us to say that the interaction between VLDLR and ApoE2 ligand binding domains with VLPs was a direct one, not requiring intermediate association with virus-like particles, and that the interaction was specific because it could be inhibited by a receptor binding antagonist that is known to bind the ligand binding domain, this RAP protein shown here. And so another step that is important to demonstrate beyond just the direct association is that this putative receptor would allow internalization of particles into cells. And so again, we relied on our trusted Keynote cells, which unless, again, overexpressing receptors, we found are non-permissive to alphavirus infection. So in this case, what I'm showing you are images obtained from live cell confocal microscopy. There's an XY slice on the left and the 3D volume on the right. In green, you're seeing wheat germ agglutinin, which is a cell surface membrane marker. And in pink, you're seeing fluorescently labeled VLPs. And so we're able to see here, if you look at the top set of images at 4 degrees, we saw what seemed to be association of VLPs to the cell surface. When we increase the temperature to 37, then we saw internalization of virus-like particles into cells. And so we were able to work with fantastic live cell confocal microscopist at Harvard Medical School and the Micron core to then quantify the number of particles that were internalized or at the cell surface. And hopefully you can see that Keynote cells were expressing VLDLR at 4 degrees, have a number of VLPs per cell shown on the y-axis bound, and at 37 degrees inside the cytoplasmic fraction, the next set of next part of the graph here under VLDLR cyto, you'll see that the particles are seemingly internalized at 37 degrees. So these are all experiments we were able to repeat with both SINV virus and Eastern equine encephalitis virus, as well as incorporating an MXRA8 control here, showing again that overexpression of this receptor set allowed both VLP binding and internalization into cells, which would be consistent with their role as bona fide cellular receptors. And so now to address the physiological relevance of that association, I mentioned to you that Semliki Forest virus causes encephalitis in neonatal mice. VLDLR and ApoE2, interestingly enough, are overexpressed on neurons during development. And so we worked with the lab of Isaac Chu to isolate cortical neurons from neonatal pups, day 0. So in this case, the Chu lab has a protocol to be able to maintain these in culture, and maybe it doesn't project well here, but you can actually see dendrites in this case between these cortical neurons. So when we incubated the neurons with no protein and then added SFV RVPs, we saw robust infection here with the no protein image shown on the top left. When we added the receptor antagonist RAP or VLDLR-LBD-Fc fusion protein, we saw inhibition of RVP infection, and we didn't see that with the isotype control. And these data now are quantified on the bar graph on the right. So there it looked like RAP or VLDLR-Fc fusion protein, so a decoy receptor, could both inhibit entry of RVPs into a physiologically relevant cell type. So we then worked with the lab of Scott Weaver to test whether or not administration of a VLDLR-LBD-Fc fusion protein could block infection and lethal encephalitis in neonatal mice infected with Semliki Forest virus. So as you can see here, hopefully, with the gray and sort of black lines here with PBS and isotype control, SFV infection of day 7 mice is uniformly lethal. Within 3 days, all mice that were challenged with SFV and treated either with PBS or isotype control rapidly succumbed to infection. But with a single administration of the VLDLR-LBD-Fc fusion protein, about 6 hours before viral challenge, we saw robust levels of protection, ultimately such that at day 3, 100% of the mice were still alive. And we still did see, sort of delayed death in this case. By day 8, all the animals that succumbed to infection. But I should add, we had not done any careful studies to look at pharmacokinetics to figure out whether or not administration of repeated doses, meaning 6 hours before infection and then repeat on day 1 and day 2 and day 3, whether or not that would have had an increased benefit of survival. But in this case, this is a very robust model that's uniformly lethal, and we did see a statistically significant effect with protection using the receptor decoy protein, which is, again, consistent with the role of this association between VLDLR and ApoE2 as a determinant of the pathophysiology of Semliki Forest virus in neonatal mice. And so taking a step back to what I was talking about in the beginning of this, my presentation, it's interesting that these viruses are able to infect these mosquito vectors, so arthropods, are able to infect birds, are able to infect horses and humans. And so the possibility that these viruses are using a lipoprotein receptor as their main way of entering cells was quite an attractive hypothesis, because several of the proteins that are, for example, important for carrying and leading to the uptake of lipids inside the gut of a mosquito are also related to the molecules that bind lipids in the serum of birds and humans and horses. So LDLR family members are quite conserved across various domains of life. And so the graduate student in the lab, Lars Clark, had the idea of cloning out VLDLR and ApoE2 orthologs from multiple species to test the hypothesis that even in these divergent organisms, these molecules could be playing a role as evolutionary conserved receptors for alphaviruses and allowing them to, again, infect a wide range of different species. And so here I'm showing you examples of experiments we did with Semliki Forest virus RVPs and Eastern equine encephalitis RVPs in red and green respectively. I'm showing you the percent of GFP positive cells when cells that overexpress human, horse, bird, Aedes aegypti or Aedes albopictus, to mosquito species of VLDLR, or even something like C. elegans, which actually initially was supposed to be our negative control because we thought the VLDLR ortholog from worm would be divergent enough from humans that perhaps it wouldn't work as a receptor. The set of receptors here are overexpressed again on Kpac62 cells, shown on the right for EVE as well. And hopefully you can see that SFV is quite promiscuous in being able to use VLDLR orthologs with organisms dating as far away from humans as the worm ortholog in this case, derived from C. elegans. While Eastern equine encephalitis virus, RVPs were able to infect human-expressing VLDLR, of course, but also the two mosquito orthologs we tested in this case, Aedes aegypti and Aedes albopictus. So we also wanted to do a similar set of experiments with A4ER2. For A4ER2, there's not a defined ortholog in mosquitoes or other invertebrates, so we only tested mouse, horse, and bird. And hopefully you could see here that there's an interesting pattern whereby SFV, again, seems to be quite promiscuous in being able to use multiple of the isoforms we tested, and EVE being able to use horse via A4ER2, and maybe less so bird and mouse in this case. And so at the time we were preparing our paper for publication, as you can see, it was a lot of work with a lot of controls, and going in back and forth with reviewers over about a year and a half. Another paper came out from Mike Diamond's group that got us really excited. This was describing the role of LDLR-83 as a cellular receptor for Venezuelan equine encephalitis virus. So this virus, like Eastern equine encephalitis virus, causes encephalitis in horses and humans, also vectored by birds, also vectored by mosquitoes. And so if you compare VLDLR and LDLR-83, this protein that is actually related to LDLR family members because it contains these repeats, but not as much because it also misses the core of the EGF beta propeller domains as well as the Oleg sugar domain. So it's really like a minimal form of the receptor. VLDLR, it's somehow LDLR-83 is involved, overexpressed in the brain, but the function and role in brain development is actually poorly characterized and unknown. But Mike Diamond's group and others ultimately, through structural analysis, were able to show that the first repeat here of this LDLR-83, so LA repeat 1, is able to bind exactly where MXRA8 binds, which is the cleft between E1 and E2. But because there's structural homology between LA repeats found in LDLR-83 and LA repeats found in the VLDLR ectodomain, we wanted to see whether or not LDLR-83 could serve as a receptor for other alphaviruses that use VLDLR. And what we saw indeed was that Semliki Forest, Eastern equine, and SINV could not use LDLR-83 despite their ability to bind the ligand-binding domains of other alphaviruses. And so ultimately, this suggested to us that perhaps there was extensive co-evolution between each of these viruses with the ligand-binding domains of their receptors, but perhaps VEEV had really co-evolved with using LDLR-83 and had become specialized with using that receptor only. So in summary, I've showed you that LDLR-related receptors are cellular receptors for multiple alphaviruses, that the receptor ligand binding domain is a site of virus attachment, that divergent receptor orthologs can support alphavirus entry, and that virus-host coevolution likely drives receptor usage patterns. And this is work that we're pursuing now in our lab to further define using structural methods. And so there are several members of the lab, both former and current, that I'd like to acknowledge. There's Lars Clark, who was a graduate student with me, but also had worked as a technician for about a year and a half before he joined the lab as a PhD student, who really was the person to spearhead this project. Chewy and Sarah. Chewy did the original CRISPR screen. He was a postdoc in the lab that's since moved on and is working now at Beam Therapeutics. And Sarah was also a graduate student who worked closely with Lars really quite an effort in terms of the number of experiments we did to really convince ourselves about the role of VLDLR and ApoE2 as receptors for these viruses. And Lars and Sarah and Shui really worked hand in hand to make this work happen. Adrian and Catherine are all other graduate students, and these are additional lab members that offered moral support. Paula Montero Lopez at the Micron core helped us with the fluorescently labeled virus-like particles. Isaac Chu's lab with the cortical neurons. I did not show you these data, but we also worked with Tracy Young-Pierce's lab at Brigham Women's Hospital to make human neurons derived from stem cells and show that RAP as well as VLDLR-Fc fusion proteins could block entry of Semliki Forest virus into human neurons. And Scott Weaver's lab at UTMP and Scott himself, and then additional collaborators at the School of Public Health. And for funding sources, really the Valley Foundation and the Burroughs Wellcome were critical to allow us to carry out this work. So I'd be happy now to take any questions you might have.
Thank you so much, Dr. Abraham, for that informative presentation. And we will now start the live Q&A of the webinar. And if you have any questions you want to ask, please do so now. Just click on that Ask a Question box located at the far left of your screen. So let's get started. It looks like we already have some great questions coming in. Dr. Abraham, are there other viruses that use LDLR-related receptors for viral entry, and do they bind the receptor in the same way?
Yes, that's a great question. So there's two viruses I can think of top of my head where the sort of important point to make is one is vesicular stomatitis virus. So this is a virus that actually, when I was a graduate student, was thought not to have a receptor because it just got into pretty much every single cell you threw the virus at. So it was thought to actually bind to phospholipids. But ultimately, a lab was able to show that this virus binds not only VLDLR, but also LDLR and other LDLR-related receptors. And that virus in particular also binds a ligand-binding domain, and kind of like the Venezuelan equine encephalitis virus structure I showed from Mike Diamond's group, they bind in a way that engages only a single repeat at a time, although they can bind multiple repeats as opposed to just one. Another example are human rhinoviruses of certain minor serogroups. Those can also bind VLDLR specifically. And so I think it's an interesting part of biology. Another point, actually, Amy Hartman's lab over at the University of Pittsburgh, I believe, showed that LRP1, which is related, but more distantly than LDLR is related to ApoE2 and VLDLR. LRP1 is a receptor for Rift Valley fever virus. And I'm waiting also, I guess, to see maybe follow-up work from that lab to see exactly how it binds. But I believe it binds a ligand-binding domain in a way that's inhibited by RAP. So a lot of viruses seem to hijack a ligand-binding domain to get into cells.
Thank you so much. And our next audience member wants to know, do alphaviruses have pandemic potential?
That's a great question. So I would argue, so there's certain properties that make certain viruses more likely to cause pandemics. I think we've all learned this from SARS-CoV-2. I think any virus that's respiratory poses challenges. Any virus also that has a ability to infect, but in a substantial number of people cause minimal to no symptoms or asymptomatic illness or mild symptoms, those viruses also have pandemic potential. So I would say for the ones that are encephalitis causing, because they're so highly lethal and people are so asymptomatic when they get sick, I think it's less likely that those viruses could cause a pandemic. That being said, viruses can always, these are RNA viruses, and they could always mutate. So for an example of this is chikungunya virus, an alphavirus that causes rash and arthritis. It did not cause a pandemic, but caused very large epidemics in recent decades, including in 2016, an outbreak in the Americas that led to over 300,000 cases. In that case, the virus causes rash, arthritis, and it's spread by mosquitoes. But another bit to mention is as soon as a virus evolves to be able to spread from human to human, that's also a game changer. So I think these are all factors to keep in mind. But for now, the pandemic potential is perhaps less than the epidemic potential, which could still be a big problem.
Thank you so much. And again, I want to thank our audience for these great questions coming in. We have one more that has come in from our audience. What makes a virus more likely to cause a pandemic?
Great. So I think touching upon these points again, I would say what we've learned from SARS-CoV-2, ultimately, if the virus is able to spread by respiratory droplets, if the virus causes asymptomatic or minimally symptomatic illness, and ultimately the virus can learn to spread human to human, those are all really big danger signs. I think, for example, the next set of viruses other than coronaviruses I'd be looking out for something like Nipah virus or henipaviruses, where in recent outbreaks there's been demonstrated human-to-human transmission by the respiratory route, but not alphaviruses just yet. These are still mosquito-borne viruses. So big pandemics, but not big epidemics, but not pandemics.
Those pesky mosquitoes. Dr. Abraham, thank you so much for your presentation today. Would you like to provide any final comments for our audience members today?
I would not have any additional comments.
Well, we want to thank you very much again for your time and for your important research. And before we go, I want to thank our audience for joining us today and for their interesting questions and questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until September 8th, 2024. Lab Roots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.